You know that fact you probably learned in high school biology?
Oh, I think I know where this is going.
Right. The one where the teacher leans against the whiteboard and tells you that you are actually more microbe than human?
Yeah, the classic 10-to-1 ratio.
Exactly. That bacterial cells outnumber your own human cells by 10 to 1.
It's basically the ultimate dinner party trivia. I mean, people love to bring it up to sound profound.
Oh, totally. It makes you feel like this, like, walk-in talking alien mothership. But, well, hold on to that feeling, but drop the math.
Right.
Because, as we're going to see today, that 10 to 1 ratio is a complete decades-old myth. And if the math we've been taught for decades is wrong, I mean, it kind of forces us to rethink everything about these invisible ecosystems living inside us.
It really does. It's a monumental shift. Because for a century, modern medicine basically treated the human body like a fortress.
Right, like we were the castle.
Exactly. We were the castle, and bacteria were this invading army that just had to be kept outside the walls at all costs. But the sources we're diving into today reveal a much more complex ecological reality.
Yeah, it's wild.
The human body isn't a fortress at all. It is a bustling, highly competitive habitat.
And before we get into what these microscopic communities are actually doing to our biology, I feel like we should probably clarify what we're even talking about.
Good idea.
Because reading through the source material, it seems like the scientific vocabulary itself has had a bit of an identity crisis over the years.
Oh, massive identity crisis. You'll hear the terms microbiota and microbiome thrown around, often interchangeably in casual conversation.
Or even in news articles.
Right, even in the news. But they refer to different things, and the distinction really matters if you want to understand how the science actually works.
Okay, let me see if I can translate this into something tangible for you listening. If we think of the human body as a theater,
Okay, I like this.
the microbiota is just the cast of actors. It is the raw census of the actual organisms, the bacteria, the fungi, the viruses present in a specific area.
Right, just who is in the room.
Exactly. But the microbiome is the entire theater production. So it's the actors, sure, but it's also the script, the stage design, the lighting, the temperature of the room.
Yeah, that's a highly accurate way to visualize it. Because Gabriel Berg actually wrote a major consensus paper in 2020 about this exact confusion.
Because scientists were just using the words however they wanted.
Pretty much. And Berg argued we needed to restore the ecosystem-level meaning of microbiome. It encompasses the organisms, their collective genetic material, and the environmental conditions of the habitat itself.
Like the pH levels and the nutrients.
Exactly. A microbiome is a dynamic, self-organizing community, not just, a bag of isolated germs.
Right. And for a long time, the medical field just used the term normal flora to describe this, right?
They did. But early microbiologists really wanted to pull the field away from the narrow, infection-centered legacy of the late 19th century.
Because back then, it was all about Koch and Pasteur and the germ theory of disease.
Right. Which was revolutionary. Identifying pathogens saved millions of lives. But it meant that for 100 years, the field largely ignored the vast majority of microbes that live with us in symbiosis.
I mean, Joshua Lederberg coined the term microbiome in 2001, specifically to shift the focus, right?
He did. He was a Nobel laureate, actually discovered bacterial conjugation. And he wanted a new framework to study the organisms providing mutual benefit rather than just the ones making us sick.
But wait, people had thought about this way before 2001, though.
Oh, absolutely.
Because I saw in the sources, Ilya Mechnikov wrote a book in 1907, The Prolongation of Life. He was literally proposing eating yogurt for the lactic acid bacteria.
Yeah, way before germ theory had even fully taken over. And Theodor Escherick was cataloging infant gut bacteria back in the 1880s.
It's crazy. Which begs the question, if these beneficial bugs have been living on us and inside us since the dawn of humanity, and people were writing about them a century ago, why did it take until the early 2000s for us to realize we're basically a walking wildlife reserve?
Well, the delay was purely technological. For most of modern history, if you wanted to study a bacterium, you had to grow it in a petri dish in a lab.
Right.
But if a microbe couldn't survive in those artificial conditions, it essentially didn't exist as science.
And I imagine a plastic dish in a brightly lit, oxygen-rich room is a very different environment than, say, the inside of a human intestine.
Radically different. Most of the bacteria in your gut are strictly anaerobic. That means oxygen is highly toxic to them.
Oh, wow.
So the moment you swab them and expose them to the air in a lab, they just die. You couldn't see who was living in the theater because they wouldn't perform on our artificial stage.
So how do you study an ecosystem that literally dies the moment you try to look at it?
You bypass the need for the organism to be alive at all.
Oh.
This required a technological revolution in DNA sequencing, specifically using something called the 16S ribosomal RNA gene. You can think of it as a universal bacterial barcode.
A barcode, meaning you just need the genetic signature, not a living specimen crawling around on a plate.
Correct. It built on Carl Woes's work from the 70s. Scientists realized they could take a sample like a swab from your skin or a stool sample, wash it to extract all the loose genetic material, and just run it through a sequencer to read those barcodes.
No culturing required.
None. Suddenly, the lights turned on in the theater. David Rollman's lab published this huge PNAS study in 2005 doing exactly this. And by the mid-2000s, this culture-independent method revealed a level of microbial diversity that Petri dishes had completely missed.
And that kicked off these massive mapping projects, right? Like the U.S. Human-Microbiome project in 2007 and the European Metatype project in 2010.
Yes, huge collaborative efforts.
And the numbers from these early surveys are staggering. Researchers cataloged over 3 million distinct microbial genes in the human gut.
Just in the gut.
Just the gut. And to put that in perspective for you, the entire human genome only has about 20,000 genes.
Right. Our own genetic complexity is completely dwarfed by our microbial passengers.
It's wild.
And what these mapping projects proved is that there is no such thing as a single, uniform human microbiome. Your body is a collection of highly structured, distinct topographical zones.
Yeah, Elizabeth Grice's research at the NIH mapped this out, and the topographical maps are fascinating. The oily ecosystem of your forehead supports a completely different community of organisms than the moist, humid environment of your armpit.
Or the dry, arid plains of your forearm.
Right, so your forehead is basically a microbial desert, and your armpit is a dense rainforest.
Exactly. And then you have the gut, which is this deep, dark, nutrient-rich,
Yeah.
oxygen-free fermentation chamber.
Sounds intense.
It is. It's dominated by these massive bacterial groups like Bacteroidetes and Firmicutes. And your specific mix in that chamber is incredibly personal.
Like a fingerprint.
It's exactly like a fingerprint. You and I share over 99.9% of our human DNA, but our gut microbiomes might only share 10 to 20% of the same microbial species.
Okay, so we are covered in these unique, highly personalized habitats, but if they're living everywhere, they're clearly consuming our resources to survive. So what are we getting out of this deal? Because they aren't just passive hitchhikers.
Far from it. They are active metabolic engines. And to really prove this, researchers use germ-free mice.
Oh, I read about this. This is fascinating.
Yeah, these are mice born and raised in completely sterile, sealed isolators. They have absolutely zero microbes of their own.
Right. And this is where the research takes a wild turn. I saw this landmark 2006 Nature study by Jeffrey Gordon's lab.
Yes, Peter Turnbull was the lead author on that.
Right, so they took these germ-free mice, which are naturally quite lean, and gave them a gut microbiota transplant from obese mice.
And what happened to the lean mice?
They rapidly gained fat. But the crucial detail, the thing that blew my mind, is that they were eating the exact same amount of food as there were before the transplant. Their diet didn't change at all.
Right, the calories in stayed the same.
Mm-hmm.
And the mechanism behind this is what makes it so profound. The microbial community from the obese mice contained bacteria that were highly efficient at fermenting complex plant fibers.
Fibers that the mouse's own stomach enzymes were completely incapable of digesting on their own.
Exactly.
So the bacteria are essentially acting as a secondary digestive system.
That is a great way to put it. The bacteria break down that indigestible fiber and turn it into short-chain fatty acids. The mouse's intestines then absorb those fatty acids. So from the exact same bowl of food, the microbes were extracting extra hidden calories and passing them directly to the host.
Here's where it gets really interesting, though, because that raises a somewhat uncomfortable philosophical question.
Oh, it definitely does.
If these microbes are extracting extra calories and fundamentally changing how we metabolize our food, who is really in charge here? Are we just walking vessels for our gut bacteria?
Well, that tension is the core of what biologists call the holobiont theory, or the hologenome theory, advanced by researchers like Lynn Margulis and the Rosenberg team.
And what does that mean, practically?
It argues that you cannot separate a host from its microbiome. Biologically, genetically, evolutionarily, you function as a single combined unit of organization, a superorganism.
And when that superorganism gets thrown out of balance, the consequences can be severe. The sources highlight a fascinating, if slightly grim, example with Clostridioides difficile, or C. diff.
Oh, C. diff, yeah. C. diff is a bacterium that causes terrible, debilitating intestinal infections. And it almost always takes over after a patient has been given a heavy course of broad-spectrum antibiotics for an unrelated issue.
Because the antibiotics act like a forest fire. They just wipe out the healthy, competing gut bacteria. But C. diff has a survival mechanism, right?
It does. It can form these resilient spores that basically wait out the chemical attack. So when the antibiotics stop, the healthy forest is gone, leaving a massive ecological vacuum. C. diff acts like an invasive weed that aggressively blooms in that empty space.
And standard antibiotics often fail to get rid of it, because every time you use them, you just clear the forest again, allowing the weed to return.
Exactly. It's a vicious cycle.
Which leads to a completely different approach to treatment: fecal microbiota transplants, or FMT. In this major 2013 medical trial in the New England Journal of Medicine, Els van Noot and her team literally infused healthy donor stool into the intestines of patients with recurrent C. diff.
They basically seeded a new forest.
Right. And the results were so overwhelmingly successful that the Medical Safety Board actually halted the trial early.
Yeah, that almost never happens.
The FMT was curing the patient so rapidly that it was deemed unethical to continue withholding the treatment from the control group.
It's a total paradigm shift. You aren't trying to poison a pathogen with chemicals. You are introducing ecological competition, so the weed has no room or resources to grow.
But, as with any major scientific breakthrough, early successes like FMT and the obesity mice studies created a massive hype cycle.
Oh, massive. The scientific community and the media got extremely excited. And when science moves that fast, it inevitably has to undergo some painful self-correction.
Which brings us right back to that 10-to-1 ratio myth from the beginning. How does the entire scientific establishment just parrot a completely unverified number for 40 years?
I know, it's wild. It is a classic cautionary tale of scientific whisper down the lane.
Seriously.
That 10-1 figure traces back to a back-of-the-napkin estimate made in 1972 by a researcher named Thomas Luckey. He took a rough estimate of bacteria in 1 gram of feces and simply multiplied it by the total volume of the human digestive tract.
And everyone just accepted it.
It sounded plausible, so it ended up in a paper, which was cited by another paper, and soon it was in every biology textbook on Earth.
Until a team of researchers, Sender, Fuchs, and Milo, finally sat down in 2016 and carefully recalculated the actual cellular volumes and densities in a standard 70-kilogram male body.
Right, they published in PLOS Biology.
Right.
And their recalculations showed that the ratio is actually about 1 to 1. You have roughly 38 trillion bacterial cells and 30 trillion human cells. It is essentially a 50-50 split.
I mean, still incredible.
Yes.
But a humbling reminder that science is a process of constant refinement. And we saw a very similar correction happen with the sterile womb debate.
We did. So for generations, the consensus was that a fetus develops in a completely sterile environment. Then, around 2014, a wave of new studies used DNA sequencing on placental tissue and claimed to find a placental microbiome.
Right, challenging the whole sterile womb idea. It made international headlines.
But it turned out to be an illusion caused by the technology itself.
Wait, really? How?
The issue was contamination. Sequencing machines are incredibly sensitive. When you are sequencing samples with extremely low biological mass, like the inside of a placenta, even the tiniest microscopic speck of bacterial DNA floating in the laboratory air or hiding in the chemical reagents will get amplified and show up in the data.
Oh, wow. So it was just a false positive.
Essentially, yes. A rigorous 2019 Nature study by Degoffo proved it was mostly just contamination. A robust placental microbiome simply does not exist.
The field also had to walk back the enterotypes hype. There was a major 2011 paper by Arumugam suggesting that every human being's gut microbiome fell into one of 3 distinct categories, almost like blood types.
Right, the Bacteroids, Prevotella, and Ruminococcus buckets.
People thought we could start prescribing diets based on your enterotype bucket.
But biology is rarely that tidy. Subsequent research showed it's not 3 neat buckets. It's a continuous, messy gradient that shifts constantly based on your diet, your environment, and your lifestyle.
Because the early hope was that we would find a taxonomic core, right? A specific list of bacterial species that every healthy human shares.
Exactly. But my healthy gut looks completely different from your healthy gut.
Which had to be a frustrating realization for researchers.
It was. But it turns out we don't share a taxonomic core. We share a functional core. We might harbor completely different species of bacteria, but those different species carry the exact same genetic tools to perform the same metabolic jobs.
Like breaking down specific fibers.
Exactly. It's the same play being performed by different actors.
But the hype wasn't just confined to academic debates. It had real-world consequences, especially regarding fecal transplants. Because once the public heard you could potentially cure diseases or alter your metabolism by transferring dead bugs, the unregulated DIY community got involved.
With tragic, entirely predictable results.
Sure.
The FDA actually had to issue severe safety warnings in 2019 after a patient died.
Yeah, from an unscreened, unregulated at-home stool transplant that contained a drug-resistant strain of E. coli.
It's extremely dangerous.
And there are also profound unknowns regarding what else you might be transplanting. I saw a 2015 case report by Alang and Kelly. A woman successfully cured her C. diff using an FMT, but her donor was significantly overweight.
And in the months following the procedure, the recipient developed new-onset obesity, despite never having struggled with her weight before.
It's just wild. It highlights the massive risks of transplanting an entire complex ecosystem when we don't fully understand all the variables yet. You aren't just transferring a cure, you are transferring a metabolic phenotype.
Exactly. Which is a really good transition to clear up a major misconception I see all the time regarding probiotics.
Oh, yes. The marketing makes it seem like taking a daily, over-the-counter probiotic capsule is this magic reset button for your gut health.
I know. But the evidence for most commercial, off-the-shelf probiotics is surprisingly weak. The issue goes back to our ecological metaphor. If your gut is an established, dense rainforest with trillions of entrenched organisms, dropping a few billion probiotic bacteria into the mix is like dropping a single house cat into the Amazon and expecting it to take over.
They just get completely outcompeted.
Usually, yes. Their effects are narrow and temporary. They might provide some beneficial chemicals while passing through, but they rarely set up permanent colonization. Plus, we often forget that the microbiome isn't just bacteria.
Right. The theater has a diverse cast. The sources mention archaea, which are single-celled organisms like Methanobrevibacter that produce methane in the gut.
We also have a vast fungal community known as the microbiome. We have protozoa.
And we have trillions of bacteriophages, which are viruses that specifically hunt and kill bacteria. It is a highly interactive, microscopic zoo.
So, understanding all this complexity, the most vital question for you, listening right now, is how this actually affects your everyday life.
Right. What does this mean for your daily choices? It means that every time you sit down for a meal or take a medication, you are actively managing a wildlife reserve.
Let's look at diet. The typical Western diet, which is heavily processed and starved of complex plant fibers, is essentially a starvation diet for your microbiome.
Because when you don't feed the diverse species in your gut, they die off, reducing your overall microbial diversity.
Yes. This is where prebiotics and postbiotics come in.
Can you break those down?
Sure. Prebiotics are simply the complex fibers in foods like garlic, onions, and whole grains that you cannot digest, but your microbes can. They are the food.
And postbiotics?
Postbiotics are the chemical rewards, like those short-chain fatty acids we mentioned earlier, that the microbes produce for you after they ferment the prebiotics.
And the importance of feeding these microbes starts on day one of a human life. The research on early development is incredible. An infant's initial microbiome is heavily shaped by their mode of delivery, whether born vaginally or by C-section, because that dictates their first major exposure to bacteria.
But the most brilliant evolutionary mechanism has to do with breast milk.
This blew my mind.
Human breast milk contains a high concentration of complex carbohydrates called human milk oligosaccharides, or HMOs.
And the human infant completely lacks the enzymes to digest them. So the mother's body is literally expending massive amounts of energy to synthesize these complex sugars, and they pass completely undigested through the baby's stomach.
Because they aren't food for the baby.
No way.
They're specifically designed to arrive intact in the large intestine to feed highly beneficial immune-training bacteria, like Bifidobacterium. Nature recognizes how vital these microbes are for survival, so it built a targeted food source for them right into the mother's milk.
And that early colonization is critical for training the immune system, which ties into something called the hygiene hypothesis, right? Or David Strachan's 1989 work, which Graham Rook later refined into the old friends hypothesis.
Right. This is the theory that human beings co-evolved with a specific set of microbes, our old friends, found in soil, untreated water, and the natural environment. Our immune systems expect to encounter them.
They act as sparring partners that teach our immune cells what is dangerous and what is harmless.
Exactly.
Yeah.
But in our modern, hyper-sanitized urban environments, we are simply too clean. We lack exposure to these old friends.
And without that microbial training, the immune system gets bored and paranoid. It starts overreacting to harmless things like pollen or even attacking the body's own cells, which may help explain the modern rise in allergies and autoimmune disorders.
It seems like every system in the body is tied to this, even the brain. The sources outline the gut-brain axis, which is a two-way communication network between your intestines and your brain.
It operates via the vagus nerve, right? And the chemical metabolites produced by your microbes?
Yes, so your gut bacteria are essentially sending chemical text messages to your brain, actively influencing your mood, your stress levels, and your cognitive function.
Wow.
Think about the implications of that the next time you are prescribed a broad-spectrum antibiotic. You aren't just killing a pathogen, you are disrupting the ecosystem that digests your food, trains your immune system, and potentially regulates your mood.
It is a lot to process. Let's take a breath and retrace the path we just walked through the source material. We started by shedding the myth of the 10-to-1 ratio, grounding ourselves in the reality that we are a 50-50 split of human to microbe.
Right.
We explored how DNA barcode technology allowed us to map the unseen deserts and rainforests on our skin and in our guts. We saw how these invisible organisms function as metabolic engines, extracting calories, defending against invasive infections like C. diff, and actively shaping our immune systems.
It fundamentally changes how we view ourselves as biological entities. And we connect this to the bigger picture. It suggests a really provocative thought regarding the future of healthcare.
How do you mean?
Well, if our bodies are essentially complex, interconnected habitats rather than isolated fortresses, the future of medicine might not look like traditional pharmacology.
Okay, so what would it look like?
Instead of constantly developing broad-spectrum chemical weapons to scorch the earth and kill invaders, the future of healing might require a completely different skill set. It might look much more like wildlife conservation, forestry, and environmental management, applied directly to the human body, cultivating the soil rather than salting the earth.
We will all need to become park rangers for our international parks. That is a brilliant way to frame it. Thank you for joining us on this deep dive into the microscopic worlds we carry with us. Keep exploring the research, keep questioning the consensus, and next time you feel completely alone in the world, just remember, you are 50% microbe. You are a walking, talking, breathing ecosystem. You are quite literally never walking alone.